So that's the topic of chapter 14, is basically the immune system response in space and time. And in particular, this is going to focus mostly on the adaptive immune system and not so much the innate immune system, although there is a section in here on innate immunity. So we start off looking at some endothelial cells in this particular chapter, and these endothelial cells have long, kind of elongated, flattened out shape, and these form the walls of your blood vessels. And that's an important part of this understanding of how the immune system and an immune response actually plays out. Because remember, these are kind of the highways that connect the immune system to the site of infection from the lymph nodes or the spleen. So the first thing that we have to understand is what the immune cells in healthy tissue looks like, what homeostasis looks like. We did this for the gut immunity. We talked about how the homeostasis played out mostly with IL-10, TGF-beta, immunoglobulin A, IgA, and then how that shifts in response to an active infection. And we're going to take basically the same strategy here. So about 30 minutes after they enter the bloodstream, about half of mature naive lymphocytes travel directly to the spleen, where they browse for about 5 hours. And this is part of our normal homeostasis and part of our normal systems of lymphocyte recirculation. The lymphocytes are constantly scanning for antigen to which they can respond. So how the immune cells move into and out of tissues during the innate and adaptive immune responses is still an area of intense study, but we've got a reasonably good understanding of how they actually get out of the blood vessels and into these other tissues. And these lymphocytes circulate continuously through the secondary lymphoid tissues. So again, lymph nodes and spleen, we've been talking about those all semester. What we're talking about is really how they transit from the bone marrow and the thymus through the circulatory system in the heart, which is kind of at the center of this diagram, and then out to the lymph nodes and spleen. They will eventually re-enter the circulatory system through lymphatic flow, and which empties back into the blood vessels and eventually back into the heart and gets recirculated back through the body again. So, cells trafficking through a resting lymph node. The organization of lymphocytes within these secondary lymphoid tissues really is important for increasing the chances that these T cells and B cells are going to encounter their cognate antigen. So what we've actually got here are a bunch of follicles in a lymph node. So these are typically B cell rich environments. What you've got are also a bunch of follicular dendritic cells present in this one. You've got some migrating dendritic cells coming in through the afferent lymphatic system, and there's two lymphatic systems, one that comes into the lymph node, that is the afferent lymphatic, and then when they exit through the lymph fluid, that's the efferent lymphatic vessels. So afferent is incoming, efferent is exiting. If you need a way to remember that, efferent is exit. They both start with. You've got scattered macrophages that live in these zones around those B cell follicles, and you've got this whole area in here that's very T cell rich that kind of borders along the outer edges of these. There's these specialized regions of endothelial cells called the high endothelial venules, which are present, or HEVs, that are present in lymph nodes, and these are not present in the spleen. So that's over here, one of the high endothelial venules, and you've got some of those B cell follicles with a bunch of dendritic cells present in some of them. Afferent lymphatics coming into the lymph node. Cells will migrate around in these lymph nodes for a while and then exit through the efferent lymphatic vessels. They can also enter from the blood vessels, and they come in through the arteries and veins, and they can actually then migrate out of those tissues and into B cells will go to the B cell follicles. T cells will go to the T cell zones. So that's these two little arrows right here. As B cells exit the blood vessels and T cells exit the blood vessels, they enter into different regions of the lymph node. This is largely in response to a couple chemokine cues. So in particular, CXCL13 is responsible for attracting B cells into the B cell follicles and out of the bloodstream. And for the T cells, it's CCL19 and CCL21 that are expressed, and that drives migration of the T cells into the T cell zone of the spleen, of the lymph nodes and spleen. Those high endothelial venules, I kind of want to draw that a little bit just because I don't really see a good illustration of that, and I want one. So what you've normally got in your endothelial cells are these relatively flattened out rectangular sort of or oval shaped sort of cells. And then as you get closer to these high endothelial venules, these start to change shape and become gradually more square and cuboidal. And these square cuboidal regions form regions where the cells are going to preferentially migrate through the blood vessels and into the lymph nodes. Where the cells actually end up going is determined by the surface receptors that are present on the B cells and the T cells, and they're going to express different receptors. That kind of makes sense. They're responding to different chemical cues. They need different receptors to sense and respond to those different chemical cues, the CXCL13 versus the CCL19 and CCL21. So among other things, there's also this network of fibroblastic reticular cells, FRCs, that set up these sort of conduits and pathways. And these are commonly used. There's an example of a couple high endothelial venules, HEVs, that are surrounded by blood vessels and part of the blood vessel. And these kind of branch off into these HEVs that give rise to the FRC, the fibroblastic reticular cell network. And within that, you've got usually dendritic cells and macrophages clustered around. And this is an area where the T cells in particular are going to browse for antigen. This is also where the antigen presenting cells, like your migratory dendritic cells, they're going to come in and move along these sort of pathways. These are sort of like roadways that get laid out in the spleen and in the lymph nodes. And they are present in both tissues, unlike the HEVs. So lymphocytes exit the blood through this process called extravasation, and this occurs at those high endothelial venules that I just sort of roughly sketched. This is driven by adhesion molecule interactions. So normally blood flows through your blood vessels nice and smooth, and the blood cells just keep going. But eventually, when you get to these high endothelial venules, you need some of those blood cells to be able to stick to the insides of your blood vessel walls. So the high endothelial venules express some particular ligands for L-selectin, known as an addressin. This is basically an address. This says, this is a high endothelial venule, please exit the bloodstream here. This is what tells your T cells and B cells where to exit the bloodstream and how to enter into the lymph node. L-selectin is present on the naive lymphocytes. So we see this particularly on lymphocytes that have not yet been exposed to antigen. And this is one of the major molecules that's responsible for helping the cells to slow their roll through the bloodstream. They will continue to slow down and attach more and more progressively to the blood vessel walls. Further interactions with other adhesion molecules are going to stop the cells and allow them to kind of squeeze through the junctions between the cells. This whole process is basically the process of extravasation. So CD62 ligand on lymphocytes binds to ligands like the L-selectin on the high endothelial venules. This forms your addressin, which basically is just a collection of adhesion molecules that instruct lymphocytes to exit at these high endothelial venules. you've got basically some electron microscopy of some leukocytes that are in a blood vessel and trying to extravasate through the walls so that they actually get into the tissues and the spaces beyond the blood vessel walls. You can sort of barely see the outline of the blood vessel wall in this particular image. You've also got some fluorescence microscopy, which actually show that the high endothelial venules, they're a little bit enlarged, and the walls of these are a little bit thickened. So you can actually see that cuboidal shape in this diagram that they've got going on over here. And those endothelial cells, as they sort of thicken, make the walls of this blood vessel look a little bit thicker and enlarged in that area. And this is, let's see, I didn't leave myself a note on what the fluorescence staining was on this. So, but what you're looking at are capillaries. This is really, this process of exiting the tissue or exiting the bloodstream into the tissues, into the lymph nodes, is really a 4-step process. So as the cells sort of get pushed through the bloodstream by normal blood flow, the first stage of this process is rolling, where the cells form brief temporary attachments with the endothelial cells lining the blood vessels. Activation by chemokines is step 2. Arrest or stopping the rolling and adhesion, this is mediated by integrin interactions with immunoglobulin family members. So the first rolling part, those temporary interactions, are selectins, and the arrest and adhesion is really driven by integrins. So 2 different classes of cell adhesion molecules that are important for this process. And then the last stage of this is diapedesis, and that refers to the process where the cells actually kind of squeeze through those tight junctions that form the walls of your blood vessels, a lot like we saw in the last chapter. We saw dendritic cells kind of extending like pseudopods, and they're part of their cell up through the spaces between the cell walls. This is the same basic idea, except the whole cell is actually going to squeeze in between some of those endothelial cells and out into the lymph node. So endothelial cells themselves are actually active participants in this process of leukocyte extravasation. They can recognize when an arrested lymphocyte has bound and send signals that loosen those tight junctions with the other endothelial cells. And this is necessary to get the blood vessels to open up and have enough space for these cells to just kind of squeeze through, which is what's sort of happening at this last final stage. So you can see that these tight junctions actually loosen and separate, and that creates a little gap between the cells, and the lymphocytes can actually just squeeze through there. So these little blue things represent the tight junction proteins, and you can see that they're separating here, and you've got a gap between those cells and your lymphocyte that has rolled, activated, and stuck is now squeezing through those cells and down into the tissue. In addition to that, it's not just that it loosens the adhesions with the neighboring endothelial cells. Those endothelial cells themselves reorganize their whole cytoskeleton and actually extend processes that help actively draw the leukocyte in between those endothelial cells. So they're active participants in this process. They're not just kind of sitting there like stationary gatekeepers. They actually help to guide those leukocytes between those endothelial cells. This whole process takes about 2 to 3 minutes. And I have a video version of this because it really helps to see this actually in motion.
Leukocytes, or white blood cells, normally circulate in the blood unattached. The presence of an infection or inflammation triggers leukocytes to move into tissue toward the foreign invader in a process called extravasation. Extravasation requires a succession of specific cell-cell contacts between leukocytes and endothelial cells lining the blood vessel. First, inflammatory signals induce endothelial cells to exocytose P-selectin and synthesize platelet activating factor, PAF, both of which appear on the cell surface. P-selectin molecules bind oligosaccharide groups at the leukocyte cell surface. Since this adhesion is weak, the leukocyte rolls along the endothelial cells. The leukocyte also contains PAF receptors and integrins. Receptor binding to PAF results in the activation of the integrins. The activated integrins are able to bind ICAM molecules constitutively expressed on the surface of endothelial cells. This causes the leukocyte to stop rolling and to tightly adhere to the endothelium. The adhered leukocyte then begins its migration between adjacent endothelial cells to reach the underlying tissue.
Okay, that video is actually generic for leukocyte extravasation into tissues and is not really specific for leukocyte entry into high endothelial venules. The molecules involved in that adhesion, especially that initial rolling step, are a little bit different. So for entry into the high endothelial venules, it's not P-selectin and PAF, it's actually CD62 ligand, CD62L, that interacts with a molecule known as glycam on the endothelial cells. And this actually is what initiates that rolling process. The second part of this, though, where the cells arrest and stop rolling and then extravasate and squeeze between the endothelial cells, that part's pretty much the same. LFA1 is one of those integrins that interacts with ICAM, intracellular adhesion molecules, on the endothelial cells, and that halts cellular rolling motions and initiates diapedesis, or passing between the cells. So I will run that video again just so that you can watch and get a better idea of the molecules involved and catch a little bit more of that this time around.
Leukocytes, or white blood cells, normally circulate in the blood unattached. The presence of an infection or inflammation triggers leukocytes to move into tissue toward the foreign invader in a process called extravasation. Extravasation requires a succession of specific cell-cell contacts between leukocytes and endothelial cells lining the blood vessel. First, inflammatory signals induce endothelial cells to exocytose P-selectin and synthesize platelet activating factor, PAF, both of which appear on the cell surface.
So that would be different.
P-selectin molecules bind oligosaccharide groups at the leukocyte cell surface. Since this adhesion is weak, the leukocyte rolls along the endothelial cells. The leukocyte also contains PAF receptors and integrins. Receptor binding to PAF results in the activation of the integrins. The activated integrins are able to bind ICAM molecules constitutively expressed on the surface of endothelial cells. This causes the leukocyte to stop rolling and to tightly adhere to the endothelium. The adhered leukocyte then begins its migration between adjacent endothelial cells to reach the underlying tissue.
So this is one of the differences between this process in an infected tissue and versus one at the high endothelial venules. The chemical messengers and signals that are actually involved are a little bit different. And what happens in the case of the active infection, because they started this video with the bacterium being shown, is that once you actually have bacterial penetration into these body cavities or into an area of the body, like say the skin, for example, the deeper layers of the skin, what happens is that triggers the endothelium of the blood vessels in that area, especially the ones that may have been damaged that allowed the bacteria in the first place. Those particular blood vessels start to express new and distinct cell surface antigens, cell surface markers that allow these leukocytes and lymphocytes to start the rolling process in the first place. Normal healthy endothelium cells do not necessarily express those same rolling kind of capture markers. So the cells would just keep going.
Leukocytes.
Yes. Nice, good, we did that. The spleen does not actually have high endothelial venules. So this is a little bit different in the spleen. Selectins do not appear to play a role. They're still trying to figure out exactly how cells do recognize that this is the site of exit into the spleen tissues and not in the absence of the high endothelial venules. But we do know that the lymphocytes do stop and they do gain entry into the spleen's white pulp via distinct homing mechanisms. And the arterioles, the incoming blood vessels that are contained, so this would be an example of one of the arterioles, those do actually contain distinct immune cell cues that trigger the extravasation of those leukocytes into these regions, and they kind of dump the cells directly into the marginal sinus. So this is the space, the marginal sinus is going to be this area right in here. So the B cells then will migrate from these central arterioles in response to CXCL13 into those follicles, these round blue spots, and the T cells will be attracted to CCL19 and CCL21 and form the T cell zones of the spleen. And we've looked at spleen architecture a number of times this semester, so this should be vaguely familiar to you all by now. So the structure of the spleen and the lymph nodes are a little bit different, and that's because they actually serve two different functions in the body. The lymph nodes really are set up to be secondary lymphoid tissue. The spleen actually does two things. It serves as a secondary lymphoid tissue. It's got all these B cell follicles and all these T cell rich zones around those B cell follicles. So it's got that same kind of architecture, but the other function of the spleen, and this is an important one from anatomy and physiology, is in the recycling of your red blood cells. So keep in mind that the primary function of your spleen is not immunogenic. It's actually for your red blood cells and recycling of those. And you can live without a spleen. It is actually, it increases your susceptibility to certain infections, especially bloodborne infections. But mostly the rest of your life just kind of goes on as normal, and there's not a lot that you have to worry about if you have to have your spleen removed. Normally that's only done if you've had significant trauma to the spleen and it's like bleeding, because it can actually make you bleed to death. All right. So these naive lymphocytes, once they come into the lymph nodes or spleen, they browse for antigen along these reticular networks in the lymph nodes. So the follicles are kind of in the red stain in this particular fluorescence image up here at the top, and the paracortex is the T cell zone. That's where you're going to see those green highlighted T cells. And then C is just kind of an enlargement. So you can actually see that the FRC network, the fibroblast reticular cell network, actually forms this mesh-like structure. And this is going to form those roadways for these T cells to kind of migrate along these distinct pathways that they're going to use to browse for antigens. So mixed in here, you're also going to have a bunch of dendritic cells. There's a few of them kind of scattered in throughout here. They're sort of in this more yellowish color. If you can actually kind of see a little bit of the difference there. So there's one there. There's probably another one there, maybe one over here. Might be one up in there as well. It's kind of hard to tell for sure. So on the left-hand side of this, you've really got the T cell and B cell rich areas of the lymph nodes, and you can see that the lymph nodes, like what we've been drawing this semester or what we've seen in the diagrams this semester, these sort of oval regions, that really is what they look like when you actually take a tissue section and you stain them for these particular types of cells. The B cells really do form these sort of oval-ish kinds of clusters that are surrounded by T cell rich zones known as the paracortex of the spleen, of the lymph nodes. So this chapter, I actually have a lot of videos, which was really exciting, although it took me forever to download all of these from your textbook and stick them into the PowerPoint. But I think it's important and interesting to actually get to see these processes like playing out really in vivo, and to actually, because this is done in living animals and not just as cell culture dishes. This one is 2-photon imaging, so a particular fluorescence microscopy technique of live T and B cells within a mouse lymph node. So you can actually see the motions of the T cells and the B cells in this lymph node of the mouse. And they kind of look like they're just swimming around randomly. But they're actually following those fibroblast reticular cell network pathways that are laid out in the structure of the spleen. So what this tells us is that once the cells actually exit the high endothelial venules into the lymph nodes, they don't just like park. They actually swim around and move around in the spleen or in the lymph nodes. Usually this one's in particular in the lymph nodes. They actually move around, and that's important because what they're doing is they're constantly sampling antigen from antigen-presenting cells. Those antigen-presenting cells are a little bit more likely to be stationary and move around a little bit less. So that those T cells and B cells can kind of browse for antigen and look for their cognate antigen. Yes, good. So we do see antigen-presenting cells present in all the lymph node microenvironments. So in the subcapsular region, you've got dendritic cells surrounded by macrophage in green here that show up, that are surrounded by macrophages in red. There's a whole ton of macrophages that are present in that particular one. In the paracortex region, in the T cell zones, you've got dendritic cells in green interacting with red T cells. So a few of those are scattered in there, and you can see that the T cells are really outnumbered. There's a ton of dendritic cells that are actually present in this particular image, and the T cells are significantly fewer in number. So there's plenty of antigens and antigen presentation going on for these T cells to kind of migrate around and browse and look at and see if they recognize any of those particular antigens. And even in the B cell follicle, we see some dendritic cells in the B cell follicles as well. Again, those green cells with the blue in this particular case, it's a follicle marker for the B cells. So we see these antigen presenting cells really present throughout the secondary lymphoid tissues. And another video. So in this particular one, the dendritic cells, which are green, are going to travel along a bed of more sessile, stationary, long-term resident macrophages, which are all the red cells. You can tell that the dendritic cells are vastly outnumbered. This is generally true. There's more macrophages in your bloodstream than there are dendritic cells. So this is not unusual. Early estimates with static imaging, if you just took this picture, what you would get the general idea of is that up to 500 T cells probed the surface of a single dendritic cell per hour, but dynamic imaging, like this in vivo video stuff that we can do now, showed that to be a gross underestimate. The movements of these T cells was a lot faster than we expected, and the actual real number is about 5,000 T cells per hour can actually survey a single dendritic cell. 5,000 T cells per hour. I can't do anything at a rate of 5,000 per hour. But what that does and what that tells us is that it's easier for us to understand how a dendritic cell with its particular antigen encounters the right T cell with its particular MHC and receptor and T cell receptor. And that recognition can actually play out. Naive B cells spend a similar amount of time scanning for antigen, but travel on distinct networks within the follicle of the lymph node or spleen, so they're a little bit different, but not much. It's the same basic idea. So you can see that those dendritic cells just kind of like crawl around a little bit in the network of much more stationary macrophages. But this movement that they do, this swimming motion that these leukocytes and lymphocytes do in the spleen and in the lymph nodes, is really basically, it's not swimming. They don't have cilia or flagella or anything like that. They're just extending pseudopods in particular directions and then pulling themselves along in those directions. So I kind of refer to it as swimming because that's what it looks like to me, but the mechanism for that really is pseudopod based. Yes, good. Okay, so again, the polarization and the movement of these lymphocytes in these lymph nodes in particular, and then also in the spleen, is going to be driven by the cytokine and chemokine cues that are present. So what you've got here is one of those high endothelial venules, kind of down here, labeled HEV. You've got two of them present in this diagram. So one here and one here. And you've got, in this particular case, a naive T cell that migrates out of that upper high endothelial venule, and then B cells migrating out of this lower endothelial venule. So we're definitely in a lymph node. So T cells coming out here, B cells coming out here, and then these cells are going to migrate along these pathways, these fibroblast reticular cell network pathways, in response to CCL19 and CCL21 for T cells, and CXCL13 for B cells. And the receptors for those particular chemokines are CCR7 receptor, CC receptor 7 on your T cells, and then CXC receptor number 5, CXCR5 on your B cells. And this is what allows those cells to sense and respond to those chemokine cues and migrate into the correct location for those particular cells and those particular sub-regions of the lymph node. In this particular inset image down here in panel B, what you've got are some of the pathways that the cells that are actually taken being highlighted. So they tracked the movements of individual cells. Like this red one kind of took this squiggly pathway and ended up over here. Another cell took this blue pathway and migrated over there. And then there's the green pathway and the yellow pathway and this pinkish purplish pathway as well. So all of these different pathways that some of those lymphocytes took as they exited from this portal area at the high endothelial venule and started to migrate through those tissues. As these lymphocytes move, they along these fibroblast reticular cell network, the FRC conduits, they're also browsing for antigens. So it's studied all along these reticular cell networks are dendritic cells that are kind of pictured tucked into kind of the corners and along the lining of these FRC networks. So these are antigen presenting cells that are shown in here. Giving and fibroblasts, actually, your textbook has them labeled as FRC cells, fibroblast reticular cells that actually give off these cytokines and chemokines. My bad. I thought those were actually the dendritic cells. They don't actually have the dendritic cells that are present along these networks kind of pictured in here, but they are present. So if there's no actual infection, or if that particular antigen isn't found in that specific lymph node, these cells upregulate the S1P receptor. And we've seen this S1P receptor before. This is not new to this particular chapter. This is basically kind of an exit signal, and this allows these cells then to exit the lymph node via the efferent exit lymphatics in the medullary sinus. The cells leave via portals and re-enter the circulation. And so this image actually shows traces of these lymphocytes as they exit. So again, you've got the portal area and different pathways that these particular cells are taking as they exit from these regions of the lymph nodes. They all kind of congregate to this particular portal area, and then they exit or dump into the efferent lymphatic vessels and then those cells exit and circulate through the body and into the bloodstream, where they will go find another lymph node to browse and see if that one shows any signs of an infection. And repeat. So this particular video is of lymph nodes exiting the lymph nodes through the, or lymphocytes exiting the lymph nodes through the portals. And the little yellow dots kind of show the pathway, or the little follow dots show the pathways of cells. And you'll notice that they disappear. And that's as they actually leave. Bloop. They really do look like they're just swimming around in there. But they're actually following some of those. You'll notice that they're associated with that red stained stuff. That's the fibroblast reticular cell network, the FRC fibers that are present. And a lot of the cells are kind of associated with those particular fibers. I think it's so cool that we can actually see the movements of cells in living organisms at this point. Like, we've got the technology to actually do that. When I was coming up in immunology, what I got were static pictures. Like, that was the extent of our technology, and very little of it was actually fluorescence microscopy. Mostly what I got was black and white pictures. So it wasn't nearly as colorful or as pretty or as easy to see and understand. And so some of these like intravital imaging techniques that we've got now, where you can do them in a whole mouse, they've really revolutionized our understanding of how an immune response plays out and how these cells actually move and respond to different kinds of cues. And it's really mind-blowing, and it's cool. I like it. And I'm really glad that at least in this chapter there were a bunch of videos to kind of work with. Okay, so that's kind of the homeostasis of what goes on in the normal body during a non-immune response. So your cells are constantly circulating throughout your body. All of these T cells and B cells are circulating throughout your body, browsing the various lymph nodes, exiting at the high endothelial venules, entering into the spleen or the lymph nodes, depending on which tissue we're talking about. They browse for antigen along those fibroblastic reticular cell networks. If they don't find it in about 5 hours or so, they'll upregulate S1P receptor, and they'll exit in through the lymphatic system, and they'll go off to another lymph node and try it again. And they'll keep doing that over and over and over again, and they'll just continuously browse until they do find their cognate antigen, or until they wear out and they die. Those are basically kind of your two fates. Something smell burny? Maybe it's, yes? That's weird. Okay. I don't see anything. The smoke alarms aren't going off. I guess we're okay. So the next part of this chapter looks at the innate immune response to the beginnings of like what happens when you first get an infection in the body. How does that innate response kind of play out? What are the initial stages and steps in that particular process? And this really begins with like basically pattern recognition receptors, going back to way earlier in this semester, recognizing your pathogens. So these are things like the toll-like receptors that bind to pathogens via the pattern recognition receptors, the toll-like receptors, the nod-like receptors, the C-type lectin receptors in some cases, particularly for fungi in that case. This pattern recognition receptor binding generates signals that release inflammatory signals, including cytokines and chemokines. And again, those affect the blood vessels and the tissues. So these initial early signals, when a bacterium actually or a virus actually penetrates into the body and it gets picked up on by these pattern recognition receptors, remember, those are present on all your cells, not just your immune cells, but all your cells. So your skin cells can actually sense when they've actually got an infection going on because they can directly detect the bacteria. And once they do, that's going to trigger cell signaling pathways within those cells that are going to tell the cells to start modulating their behavior and releasing some early cytokines and chemokines that can affect those local blood vessels in the area and assist with that extravasation of leukocytes into the infected or inflamed tissues. Chemokines attract those innate cells to the site of infection, and that neutrophils are your first responders to infection. They produce more chemokines that continue to attract other types of antigen-presenting cells, such as macrophages and dendritic cells, which are usually the second types of cells that are on the scene. So as a particular example of this, you've got the gram-negative bacteria Salmonella, which binds to toll-like receptor 4, which is expressed by enterocytes of the intestinal epithelium. Herpes simplex virus, which is a different kind of pathogen, binds to both external toll-like receptor 2 and internal toll-like receptor 9 receptors. Staph aureus, which is a normal skin bacterium, is an extracellular opportunistic pathogen that typically gains entry via the skin, and it usually is recognized by toll-like receptor 2 on dendritic cells or on dendritic cells in the dermal tissue in the skin itself. So these different kinds of pathogens get picked up on by different types of toll-like receptors and different types of pattern recognition receptors. In response to that, you're going to get different cell signaling pathways activated, which triggers different cytokines and chemokines. However, the first events are usually recruitment of neutrophils into the area, because neutrophils are really good at doing netosis. They are phagocytic, so they will attempt to engulf the pathogen, and they will help to clean up the initial infection, hopefully without getting the rest of the immune system involved. I mean, in an ideal world, the innate immune system should be sufficient to clear your infection before it can gain a foothold in the body. So what you've actually got are neutrophils and monocytes that migrate to a site of inflammation. You've got this little blue circular area up here, and this takes like over the next 20 minutes in, you start to see some migration of neutrophils in red and monocytes in green. This one is not particularly in video form, although I think the next slide is a video showing neutrophils swarming to a site of infection. But you can see that you've got a ton of neutrophils, like lots and lots of neutrophils, because lots and lots of red in these top panels, and then the monocytes in green. And then if you track the movements of individual cells, you end up with these pathways that get highlighted, and that's the bottom part of this particular set of panels. They tracked both the neutrophils in blue and the monocytes in yellow, or macrophages in yellow. And you can see that they migrate in toward that blue circle area that is initially present. So within 20 minutes, you get this like little, almost like a clot, this cluster that forms. And by 2 hours later, that's really enlarged. And you've got not just neutrophils, which were there within the first 20 minutes, but within 2 hours, you've got a bunch of monocyte macrophages that are present at that particular area as well. So this is where the antigen or the pathogen occurs. You can see that it gets swarmed by the neutrophils first, and then later those monocytes in green come into the area and attack as well. So yes, I thought I had a video of this. So what you're actually going to get to see in this one is the red neutrophils swarming to a site of damage in the ear of a mouse. What they probably did was what's called a hole punch. They take it's like a little paper hole punch, but it's considerably smaller. It's only maybe 3 or 4 millimeters across, and it punches a little hole through the ear of the mouse. The ear is a really thin tissue. It hurts a little bit. I'm not going to lie. Like, they're not happy to have this done, but it doesn't cause major permanent damage, and there aren't a lot of nerve endings in the ears of the mice, so it's not hugely painful to them. It's probably no worse than getting your ears pierced as a human. So there is that. If you're a nice researcher, you numb the area first with some ice, usually, which is often what they do for humans when they pierce our ears too. So you can actually see all of those neutrophils swarm literally to the central area of this particular slide and form this big round ball of cells. And much more slowly, you can actually, over the 2-hour period or roughly 2-hour period, you can see those monocytes and macrophages come into this particular cluster of cells as well and really form that. I mean, look at how quickly they swarm. So neutrophils are really, really aggressive cells, and they are pretty much always the first responders to any particular infection, whether it's bacterial or viral or fungal. And their job is to really surround that invader and try to eat it, try to do netosis and trap it and kill it. And then what they can't clean up, the macrophages come in and they try to eat the pathogen as well. These are literally within the first 20 minutes of a pathogen entering your body. So these are some of the first events that actually unfold, whether you're talking about bacteria or viruses getting into your system. That antigen then actually has to get to your secondary lymphoid tissue. And it's going to travel through one of two routes, depending on its size and solubility. It's got to get to the tissue either through the blood or through the lymphatic fluid. And antigen has to travel to these lymph nodes or the spleen in two different forms. One as whole unprocessed antigen for B cells, and also as processed antigen presented in MHC for the T cells. So the B cells bind to unprocessed antigen present in your lymph nodes, and the T cells scan the lymph nodes for antigen in MHC. We talked about the structure of MHC. We talked about MHC1 versus MHC2 and open and closed kinds of beds for the antigen and the size differences between those peptides and the endogenous and exogenous pathways for how those antigens actually get put into those. We talked about how B cells acquire their antigen, which is usually by stealing it from an antigen-presenting cell or through direct sampling of their environment through penocytosis for small things. So this is literally some of those pathways and where those become important for our immune responses. So up here at the top, you've got a splinter or a little piece of glass or something that punctures through the skin, through the epidermis and into the dermis. It's covered with infectious material. It's covered with antigens, pathogens of some sort. It could be bacterial, could be viral. This diagram doesn't really make that clear, and it doesn't really matter at this stage. But this gets picked up on by antigen-presenting cells, particularly dendritic cells in the epidermis and dermis regions. These migratory dendritic cells then actually migrate into the nearest lymph vessels, where they're going to travel through the lymphatic system, and they're going to enter the lymph nodes themselves, the nearest lymph node, the draining lymph node, through not the high endothelial venules, because they're in the lymphatic system, but through the afferent lymphatics. The process for presentation of antigen for B cells roughly plays out the same way. So you've still got this initial puncture injury. You still got the dendritic cells picking up on that. You still got them entering through the afferent lymphatics. The difference here is where they migrate in terms of their location and sublocation in the B cell follicles. And you'll notice you've got some macrophages that are present in the marginal zone of the spleen, or the near the B cell zones of the follicles. That's these cells over here. And so your dendritic cell can actually give some of its antigen to these particular macrophages. And some of that antigen is coming in as whole unprocessed antigen, so it's just separate. It's usually coated with complement at this point, hopefully coated with complement, maybe even coated with some antibodies like IgM or IgA, depending on which tissue you're in. And that gets picked up on, and then that can be used to transmit information to the B cells, either directly through the dendritic cells, through the macrophages themselves, or directly sampled from the environment. They did some kind of cool research with this, looking at malarial parasites. And these actually confirmed that whole pathogens can actually travel to the lymph nodes and do so within minutes of injection into the bloodstream. So minutes after injection into the skin by a mosquito bite, sporozoites that carry malaria from the mosquito travel directly to the draining lymph node. So the whole antigen is available as sporozoites for B cells, or resident antigen presenting cells can process and present that antigen to T cells as well. So what you've got here is really the same exact thing going on. You've got the mosquito that punctures the skin. This is done so that it can get a nice blood meal because it's probably a female mosquito looking to lay eggs. But the sporozoites that actually transmit malaria enter into the bloodstream through the blood vessels and the lymphatic vessels and travel directly to the draining lymph node that is going to be near the site of infection. So if you get bitten on your arm, that's going to be your axillary lymph nodes in your armpit, just as an example. And they actually got live imaging of this, which is cool. So the malarial parasite, they actually found it in a lymph node. So what you've got in this particular case is going to be some green CD11C positive cells. That means they're dendritic cells that express CD11C. Some red, that's going to be one little particular red worm-looking thing. That's going to be your Plasmodium burgii, which causes a rodent form of malaria. And then blue is just a lymphatic tracer so that you can actually see where the lymph fluid is actually moving and where it's actually going. This is isolated from the popliteal lymph nodes 3 hours after immunization. So it's not an immediate reaction. It takes a little while for the sporozoites and for the whole pathogen to actually get into the lymph nodes, but this shows that it does get there. So they've actually got the little parasite highlighted in that little circular inset, and then they zoomed in on that so that we can actually see it. So this really just shows us that it is actually possible for this whole pathogen to actually travel to the lymph nodes and actually present its antigens directly to B cells that are present or dendritic cells that are present in this particular tissue. So the antigen presenting cells activated by pattern recognition receptor signaling, this not only enhances their antigen presenting capabilities, but it also upregulates the expression of distinct chemokine receptors. So that's going to allow them to home to different tissues, again, within these particular structures within the lymph nodes. So there's a CCL21, which enables these dendritic cells to migrate to the T cell zones of the lymph nodes or the spleen so that they can do their antigen presentation role to naive T cells that are browsing for antigen. What you can see is that they kind of patrol and they move along these fibroblast reticular cell networks to basically be accessible to the T cells. They usually kind of settle along the FRC conduits in the paracortex or the T cell zone of the lymph node. The endothelial cells from the blood vessels are in particular important for expressing CCL21, which is important for antigen-presenting cell migration. If you block CCR7 and CCL21 interactions, if you prevent those from happening, the antigen-presenting cells stop crawling toward the lymph node. So this is actually a necessary set of cytokine and chemokine interactions for antigen-presenting cells to migrate along and get to the nearest lymph node. Once they get to the lymph nodes, the antigen-presenting cells also use their CCR7, receptor 7, to follow that same trail of CCL21 to the paracortex region of the lymph node. So this purplish area down here in the lymph node that's just outside of the B cell follicles that are sort of not really even labeled on this diagram, because what they were trying to show was basically that the dendritic cells migrate unless the CCL21 and CCR7 is blocked, in which case they don't actually enter into the lymph node at all. They remain kind of trapped and localized in controlling the blood vessels and the tissues. Unprocessed antigen. So this is whole antigen that has not been processed through the endogenous or exogenous pathways and presented in MHC. This is the whole antigen itself. So this retains the protein folding shapes and everything else that are present in the actual pathogen. This, if it's small and soluble, it can enter the the bloodstream. But if it's large enough, particles and whole pathogens, it tends to travel to the draining lymph nodes through the afferent lymphatics. Remember, afferent is incoming to the lymph nodes and efferent is exiting from the lymph nodes. So these small and large antigens both end up in the subcapsular sinus regions of the lymph node structure. You've got another high endothelial venule here. You've got the afferent lymphatic vessel coming in. You've got these little pink dots that represent antigen, which they've been consistent about throughout this whole textbook, which is nice. So whole antigen enters through the afferent lymphatics if it's large enough. Some of it on the surface of dendritic cells. Others of it gets picked up from dendritic cells or macrophages that are sampling that afferent lymph fluid that comes in. So this is what we're looking at over here with antigen. Some of it's on the dendritic cells, some of it's free floating in the lymphatic fluid, and some of it gets acquired by these antigen-presenting cells such as macrophages, especially the marginal zone macrophages that sample directly from this fluid, and that presents antigen then to the B cells directly for the whole unprocessed antigen. Ultimately, the B cells and macrophages relay antigen to the follicular dendritic cells, which get continuously scanned by naive lymphocytes, especially the naive B cells, but also to some extent the T cells as well, the T helper cells in this particular region. So you've got these antigens being picked up on by a follicular dendritic cell and gathered. The same basic kind of process plays out in the spleen, except you don't really have the high endothelial venules present, so those aren't there. But you've got whole antigen entering the spleen through the bloodstream. You've got the CD169 metallophilic macrophages that line the regions of the spleen that are adjacent to those B cell follicles and constantly sample the bloodstream for these particular soluble antigens. And they perform antigen-presenting functions and pass those antigens off to the follicular dendritic cell. That's what FDC stands for. I had a momentary brain fart. Some of those dendritic cells that come in through the bloodstream also extravasate into the fibroblast reticular cell network, where they can present antigen to the T cells of the T cell zones of the spleen as well. So that's two different pathways for the antigen to actually take. If it's free floating in the bloodstream, it can be picked up on by these macrophages, which can present antigen to the dendritic cell and then to the B cell. Or if it's carried in by dendritic cells, those can actually squeeze through and extravasate into these into the white pulp of the spleen. Okay. So in this particular video, we're going to be looking at, we know that complement is important for antigen transport. And one of complement's functions is supposed to be to coat your pathogen. So opsonization of your pathogen. This goes way back to the beginning of the semester. So we've got, in this case, wild-type normal mice, which are on the right-hand side of the video, and then you've got complement receptor deficient B cells on the left-hand side of this particular video. The normal B cells on the right-hand side can be seen sampling antigen from the macrophages, and they grab and run away with chunks of the red antigen. But in the complement receptor deficient cells, they can browse the antigen, but they can't acquire the antigen. So you really have to have complement receptors present on these B cells for them to be able to acquire the antigen from the environment. And that's what these videos kind of show us in real time and in real, like, living tissues. So they've got this little yellow triangle that's kind of pointing to this cluster of cells up here. And those cells, if you watch them, they start off green and then they kind of become this mix of green and red, which occasionally will show up as yellow. And I'm going to replay that video, but that little cluster of cells is going to pop up basically right in here. That little yellow triangle will show up and it's going to point to those cells. So watch what happens to these green cells in particular. They're going to pick up antigens and chunks of red from the environment. But none of the B cells in the left-hand panel actually pick up any of that antigen. They're sampling it. You can see them crawling around in the pool of antigen that's present, but you don't ever actually see them actually like rip off a hunk of antigen and acquire any antigen that way. So they don't ever actually become antigen-presenting B cells. You can see some of the like browsing up in this particular region up in here, but they don't get any of those little red chunks mixed into their cell body. So that's going to be right in this area right up in here where the little white arrow is going to pop up on that one. And I'll run this one more time. It's so neat to get to see antigen actually be picked up by cells like in video form. This is so exciting. You can tell I did my work on immunology, so I find this exciting. Yes, yes. Good, good. All right. So those blood-borne antigens are captured by specialized antigen-presenting cells at the marginal zone of the spleen. And this is still part of your innate immune response. This is still part of your non-specific immune response, but we're starting to really kind of bridge the non-specific over to the specific portions. This is how the antigens actually get from the site of infection into the tissues. And again, you've got a lymph node versus the spleen, so the slight differences in terms of the pathways between the spleen and the lymph nodes. The red pulp and the white pulp are separated by the marginal zone of the spleen. The white pulp is organized into your T cell zones, the periarteriolar lymphoid sheaths, and the B cell follicles. So in the lymph node, that was the paracortex and the B cell follicles. In the spleen, whole or particulate antigen also enters the white pulp either directly or via relay involving the CD169 positive metallophilic macrophages. So that's these guys right here that pick up on some of that blood-borne antigen that's coming in. Those are highly specialized cells. And then what they do is they kind of hand this antigen off to the FDC, the follicular dendritic cells, for antigen presentation. Now what that should do, ultimately, is start the B cells differentiating into plasma cells and germinal center cells. And in the germinal center, you should have 3 processes that go on. That would be class switch recombination, so switching from IgM to IgG or IgA or IgE, depending on the type of pathogen. You should also see the somatic hypermutation and the affinity selection that go on between the dark zone and the light zone of the germinal center once those actually do form. So this is what should kick off that particular process. So that kind of brings us to first contact between the antigen and lymphocytes. That's where we are at this particular stage of the infection. We finally get contact between the actual lymphocytes, so the antigen-specific cells, and the actual pathogen itself. What we do know is that the behavior of the T cells, B cells, and antigen-presenting cells all change dramatically during the first 48 to 50 hours, so the first roughly 2 days of a primary immune response in the lymph node. It takes a couple of days for this response to really get underway and to really go strongly and start to really produce a lot of T cells, B cells, and get this antigen presentation thing going specifically in the way that we've kind of discussed it so far this semester. So, stage 1 in the T cell zone, the paracortex of the lymph node, or the periarterial or lymphoid sheath of the spleen. You've got antigen that enters. That antigen can be picked up by a B cell receptor, or it can be processed by a dendritic cell and presented on the surface in MHC class 2, because there's your antigen on MHC class 2, where it interacts with the T cell receptor on the T cell, and that's CD4 recognizing your MHC2 for exogenous antigen. So antigen picked up on by B cell receptors for the B cells, or dendritic cells and presented through MHC class 2 and recognition through the T cell receptor and CD4. Whether we're talking about B cells or T cells, this should trigger an initial wave of proliferation in both types of cells. So we see rounds and rounds of mitosis that should occur in this first set of responses. This is antigen-mediated activation and proliferation. And then we see differentiation. And for the B cells, we're going to see plasma cells form that are going to be able to secrete antibodies. The first ones that are going to form are going to be the plasma cells that form outside the germinal center, the cells of the primary focus. And they're going to secrete mostly IgM, maybe a little bit of IgG, because they can still go through class switching. They just tend to not do it as much as cells from the germinal center. So this plasma cell probably came from one of the primary focus cells, and it secretes antibody that is bound to its, or that can then go off and bind to the particular antigen. The naive CD4 positive T cells, so your helper T cells in this particular case, stop their movements after engaging antigens. So what we see is they quit migrating around and they become really stationary, and they form these stable long-term connections with the antigen-presenting cells. The kinetics of these encounters depend on the quality, the quantity, and the availability of antigen and the dendritic cell activation state. So there's a number of factors that kind of play into how long these encounters last and how well they work. But in general terms, within minutes of binding to the MHC peptide complexes, the CD4 positive T cells slow down their movements. We saw them previously swimming kind of rapidly throughout the lymph nodes and browsing for antigen. Once they find that antigen, they stop. That's kind of the point here. And those can become involved in basically committed long-term relationships of 8 hours or more with dendritic cells. This is going to trigger them to differentiate and to divide, first of all, and proliferate, but also secondly, to differentiate into distinct helper cell subsets. Some of these are going to assist in type 1 responses, and some of them are going to assist in type 2 responses. That's going to be partially driven by the cytokine and chemokine cues that are present in the environment. And that's going to depend on which pattern recognition receptors were triggered. So cells can experience up to 10 divisions over the next 4 to 5 days that this proliferation thing is going on. So this week on Friday, I'm actually scheduled to go get my flu shot and my COVID booster. Yay, awesome. This is exactly what's going to be happening over the next few days, that I'm actually going to feel like absolute crap after having gotten both of those vaccines simultaneously, because I know I'm going to feel like crap. So far, the COVID boosters have always made me feel like crap for a couple of days, but it's usually short term. So I know what's going to be going on in my body, and I know the time frames roughly. So within the first few minutes, this process is going to kick off. But it takes about 6 hours or so before I really start to feel the effects of the vaccine. So that tells me a little bit about like when my system is starting to kind of really maximize its response. And then over the next 4 to 5 days, I'm going to be building that immunity, and those memory cells and those specific antigen presenting cells and effector T cells in particular that should be forming hopefully good memory cells so that I can fight off the infection in the future. The process is exactly the same as it was when I actually got COVID. The B cells, their behavior also changes. And remember, these need 2 signals to become fully activated, engagement with antigen and with a helper T cell. So after binding antigen, B cells upregulate CCR receptor 7. This allows them to follow the chemotactic signals that are generated in the paracortex. So these B cells are actually going to migrate from the follicle toward the T cell zone. And that's something that I think I've got a video on in the next slide. Yep, I do. What do you know? More videos. The T helper cells are also attracted to the border between the follicle and the T cell zone. So basically, all the cells, B cells and T cells alike, are going to kind of migrate to this central area along the border, along the periphery of those follicles. And this is going to maximize the interactions between the T cells and the B cells so that you can get optimal activation of both the antibody-producing cells and the helper T cells at the same time. So naive B cells express chemokine receptors such as CCR4 and CCR7 and CCR5, which attracts them to chemokines made in the follicles, but activated B cells use CCR7, and that allows them to move toward the paracortex, the T cell zone. T cells, on the other hand, are going to actually, when they're activated, they're going to upregulate CC receptor 5, which attracts them to the chemokines made in the follicle. So basically, both types of cells, when they're naive cells, that's before they encounter antigen, they're going to behave in a way that kind of keeps them separated in space in the follicles and in the paracortex, or the periarteriolar lymphoid sheath of the spleen. And then once antigen has actually been encountered, you're going to see that they start to upregulate the opposite type of receptor. So the T cells will upregulate some of those like B cell chemokine receptors and start to migrate toward the B cells. The B cells will start to migrate toward the T cells. And this dramatically increases the probability that they will actually encounter each other and deliver and receive the appropriate help signals for optimal activation. So that's what's going on in this next video. And in this video, we've got B cells in green, so you can see there's a bunch of them. One particular B cell shows some pretty dramatic movements and is highlighted with periodic pop-up circles, so you can actually follow its specific movements through this particular tissue. But overall, just kind of visually track where those green B cells end up going. So you can see them kind of like form this almost sort of a layer or a zone over here where they're more concentrated, and this is adjacent to these little red cells over here. And this, I think the red cells are T cells, but I didn't find any particular information on the staining of those, or at least I didn't leave it in my notes. But these cells actually went from being scattered and diffused throughout this region to over here in this one particular region. And I will run through that particular video again so that you can actually see it. So they start off all over the place, and then gradually they sort of migrate, and you can see that one particular B cell that just sort of like swims to this particular B cell zone, and it's already starting to take shape, and you can see that zone kind of becoming more clear at that B cell T cell boundary. Okay, this next video is actually showing the interactions between antigen-specific B cells and T cells at the border between the follicle and the paracortex. So in this case, we're supposed to be paying attention to the pairs of red and green cells, that's T cells and B cells. It's worth remembering that the B cell is in charge of this encounter. So it's the B cell that actually controls the movement of this pairing and this little dyad, this duo of cells that are attached to each other temporarily. And the T cell just sort of trails along helplessly after the B cell. How they determined that, I don't really know, other than a lot of observation that seemed to suggest that the B cell was kind of in charge here. But notice that you can frequently see areas of yellow fluorescence. So before I even start the video, you can see that there's some overlap where there's yellow going on. And in fluorescence microscopy, red plus green equals yellow. So this is what tells us that these cells are physically overlapping each other in space and probably touching each other in space. And so you can continue to see that. And there's also in this little pairing down below it, there's a little yellow spot as well on that particular cluster of cells. And so these are kind of the ones to just visually sort of keep track of as this video plays. So again, a little bit of an area of overlap in this pairing and duo there. And they go around as literal pairs of cells. And the green cells are the ones that are in charge of the movements in this in these videos. And the red cells are just sort of along for the ride. I don't know if watching these videos I would have actually gotten that impression, but that's what your textbook actually indicates. So I'm guessing there's experimental evidence that kind of shows that the B cells, like if you inhibit their movement, you don't see movement from the T cell as well. So those yellow regions of overlap, it's important to also think about what's going on in those regions. This is going to be an area where they're forming an immunological synapse. So we learned about those before. This is where we're going to have clustered T cell receptors, CD28 and CD40 ligand molecules that are going to engage with the MHC class II peptides, CD80, CD86, and CD40 molecules on the B cell. So all of this is like those molecules are what's kind of holding those cells together. So you can see that this actually forms a pretty strong and stable attachment that is able to withstand a large degree of movement. Like that's a lot of movement for little cells to be going through. And yet these connections appear to be stable and secure. So that's a pretty tight binding. Okay. We also know that B cells in the germinal center and the T cells that are in the germinal center are unusually mobile. So they actually swim around more than we expected them to when we started to observe these phenomenon in living tissues. The germinal center B cells also look really different than we thought they were going to. We figured they'd just be like round cells because that's how we always draw them and depict them as being spherical, basically. And they actually look more like dendritic cells. So I've actually got a video coming up in the next slide that's actually going to show some of these movements and the differences in morphology between these cells. The germinal center B cells also seem to sample antigen more actively and more effectively than other B cells. And so during this time, what they should be doing is acquiring their specific antigen from antigen-presenting cells by physical force, so like by stealing it, ripping it away, or acid or protease digesting it out of that cleft in the immunological synapse between those cells. So these are, again, processes that we've already learned about this semester and hopefully that you remember. So these cells spend less time in contact with T cells than we originally predicted. So when we think about what should be going on in the germinal center, you've got the light zone, which is what we think goes on there is the class switching primarily, and memory cell differentiation, and the formation of plasma cells. And then in the dark zone, you've got somatic hypermutation, and you've also got the affinity maturation that goes on and is thought to occur in the germinal center as well. So this is that process of remutating that B cell receptor after it's been formed to try to come up with an even stronger antigen match. Those follicular T helper cells help to distribute some of their help along the germinal centers, and they actually move around a surprising amount, and they will migrate and move between adjacent germinal centers. So they're not static cells either. They are constantly in motion, and this makes it possible for them to make sure that they activate as many B cells as possible, basically. Ultimately, as the cells kind of go through somatic hypermutation and affinity selection, the ones with highest affinity for antigen come to dominate the germinal center. And this is not usually just one specific thing or one specific clone of B cells. We originally kind of thought that was the case, but it turns out that it's actually multiple B cells that form a germinal center, and then you've got rounds and rounds of proliferation going on. So this ultimately kind of swells into a much bigger structure and cluster of cells that come from a number of activated B cells and not just one particular activated B cell, usually. So in this video, pay attention to the difference between the green and red cells in terms of their speed and their shape. In this one, you've got blue is collagen, which is barely visible, mostly kind of in the upper left-hand corner there, but it's just a structural stain, so you don't have to worry about it. It's not going to do anything interesting. The green are the germinal center B cells, and the red cells are naive lymphocytes, both B and T cells. So what you're looking for, again, are those differences in shape. The germinal center B cells that have become activated express a different surface marker, so that's how we're able to identify those. But even before I start the video, you can see that compared to the naive B and T cells in the periphery in the red cells, the green cells that are the germinal center B cells appear to be larger, and they also look more star-shaped or tree-shaped. Dendritic morphology is what we call that. And then we can also compare the degree of movement that these cells actually exhibit and see that they actually show more motility than the naive cells do as well. I mean, there's plenty of movement among those red B cells and T cells in addition, but the ones that are in the germinal center are exceptionally mobile. So they are moving faster and more, to me, it looks like chaotically, randomly. But again, remember, one of the things that should be happening is they should be going through the somatic hypermutation process and affinity maturation. So they should be testing themselves against a number of other types of T cells to see if they've got higher affinity for antigen. So that's probably why we see so much movement going on in these particular germinal centers that we didn't necessarily expect to see. It's really cool. And that doesn't even take into account the amount of movement that's got to happen as they kind of shuttle back and forth between the light zone and the dark zone of the germinal center to go through those rounds of proliferation and somatic hypermutation and then affinity selection, and hopefully also class switching from IgM to IgG. And all of those processes are, of course, invisible, so we don't get to see those or watch those occur in real time, but that would be cool if we found a way to do that. So this video actually is going to track the T follicular helper cells, so the T cells, and it's going to show them exiting a germinal center. The ones that are circled are going to actually leave the germinal centers. And this is, again, one of those things where they actually will move and migrate between adjacent germinal centers. So the green cells are your T cells in this particular case, and the red cells, there's actually two different shades of red, which visually I cannot tell the difference on this particular video. I'm just going to have to trust that they're different, but there's two different types of cells in red. One is going to be the follicular dendritic cells, and the other is going to be the naive B cells. Both of those are going to be in shades of red. And so these green cells you want to pay attention to are the T cells that are going to exit the germinal center. And really, we know that they kind of exit the germinal center because they actually just kind of disappear. So it kind of follows three of them. And two of them kind of seem to swim like out of the frame of view, but the one that's that migrates down toward the bottom left corner, down over this direction, I'm going to run this video again so you can watch it in particular. It actually gets to about here, and then it just kind of blinks out. It disappears. It goes away. And so it finds a little channel or escape route from the germinal center and moves on to another germinal center, we think, somewhere else in the lymph node or spleen. So it's that yellow circled one. It just kind of blinked out and is gone. Okay, so a lot like CD4 positive T cells, the CD8 positive T cells also arrest or stop their movements when they contact MHC antigen complexes on the antigen presenting cells. So they engage in both transient and stable associations. They also have a couple interesting questions, like we know that CD4 positive T cell help is required to actually activate a CD8 positive T cell. But how does that work? Because they can't directly interact with each other. Like their molecules don't match up. You can't they don't have the right kind of like MHC for antigen presentation. They don't express MHC2, so they can't express and show that antigen to their cognate other T cells. So how do they provide that help if they cannot directly engage the T cells? That's one of the big questions that took us a while to come up with an answer for. And part of that goes back to two models that we talked about previously in a previous chapter. So I'll come back to those two models in just a second. But what we've got kind of going on in this figure in the lymph node here is a high epithelial venule. So you've got that nice cluster there with the fibroblast reticular cell network and a fibroblast reticular cell kind of tucked into the corner here. And you've got two kinds of cells, the CD8 positive cells, which go off and encounter an antigen-presenting cells, the CD4 positive cells on the bottom, which go off and encounter their antigen-presenting cells, and then you've got this weird thing over here. So after that CD8 positive T cell, after it's already engaged with an antigen-presenting cell, comes over and finds another dendritic cell and forms a complex, a trio, with the CD4 positive T cell. So what we've got really is this kind of three-way interaction that goes on between this little cluster of cells that forms. And then that causes those CD8 positive T cells to divide and start to form both effector and memory cytotoxic T lymphocytes. And the CD4 positive T cells can go on and provide help to other T cells that may or may not be activated as well. So those T cells, the CD8 positive T cells, can divide up to 10 times over a 4 to 5 day period following antigen engagement, just like the CD4 positive T cells. So you're going to end up with a clone army of these particular types of T cells. So the two models that we talked about previously for CD8 positive T cell activation, so there was either sequential CD4 positive and antigen presenting cell interaction So CD4 positive cell talked to an APC, and then CD8 positive T cell talked to that same APC. So that, in particular, that would be most likely a dendritic cell for the antigen presenting cell, because they're the best at doing cross presentation. Or a simultaneous three-way interaction, which is what's being depicted both in the fluorescence image on this slide and in the diagram. So what you've got are three cells. The one shown in blue is the dendritic cell, the one shown in green is the CD8 positive T cell, and the one shown in red is the CD4 positive T cell. This is some really, really cool, awesome imaging. And I'm obviously like super excited by this because I'm a big old nerd. But I love fluorescence microscopy to start with, but the fact that we can do this in vivo and actually see this happen in a living system is really, really cool. So what we think that kind of looks like in terms of the molecules involved is the normal T cell receptor interacting with MHC class 2 on the antigen presenting cell for the CD4 positive T cell. So that's CD4 that's attaching to the MHC on the antigen presenting cell. And then you've also got another CD8 positive T cell that's kind of down below that. And it's also engaging with the antigen presenting cell through its T cell receptor and MHC1, and CD8 is recognizing that MHC1. And again, antigen in the middle. And then in addition to that, there's also a direct interaction and connection between the CD4 positive T cell and the CD8 positive T cell, and that's between CD40 and CD40 ligand. So the co-stimulatory molecules that the T cells express do provide a necessary co-stimulatory signal, and they also, the CD4 positive T cells are also a good source of interleukin-2. So are the CD8 positive T cells. They'll produce IL-2 and they will reabsorb that IL-2, and it forms a positive feedback loop on the IL-2 part. The dendritic cell also secretes a bunch of cytokines, so there's a little list there. We're not going to worry about specifically what those are, but they're all necessary to fully activate a CD8 positive T cell and get that cell to actually be able to perform its effector cytotoxic functions using perforin, granzyme, or fast ligand. So the data so far suggests that this three-way model where the cells actually form these little trios and these clusters may be the more correct way to think about this, but it doesn't rule out the possibility that the other sequential activation of a CD8 positive T cell also happens. It just is that we haven't particularly necessarily been able to track that or see that in vivo as clearly yet. So we know that these trios and these clusters form somewhere around, the one that you're looking at here was about 20 hours after T cells were exposed to antigen, so a little bit less than a day. And this video just kind of confirms, using the same coloration that we were just looking at, blue is dendritic cell, red is the CD4 positive cell, green is the CD8 positive cell. So once I play this one, you'll be able to see these three cells all together as a trio. It kind of looks like the green cell disappears a little bit, but it just sort of gets hidden by the blue dendritic cell body. This one is so cool to me. Like, I didn't think that we would ever be able to get imaging that was this detailed and this explanatory in a living tissue. So this is like mind-blowingly amazing microscopy work and immunology work to me, and I wish I had done some stuff like this in my studies, but all of my immunology work was done on like just normal old tissue slides, so static fixed tissues that were deceased already, and I just stained them for various things and looked for where those cells were, and it was nowhere near as exciting as this. This is fun. Being able to see these things actually in motion is so cool. All right, last time. Okay, so the naive cells encounter antigen. They become activated through interactions with antigen-presenting cells, particularly dendritic cells. And once those cells have become activated, they go through rounds and rounds of proliferation over a 4 to 5 day period, and then they leave. they actually exit the lymph nodes and they go off into the tissues. Depending on what kind of cell it is, that kind of determines ultimately sort of where it goes, and what kind of effector cell it becomes helps to determine its final location within the body. So plasma cells, the ones that are going to secrete antibodies, travel to different sites. The early ones in the primary focus, those tend to produce IgM and not do class switching, and they move to the medulla of the lymph node, where they're just going to secrete IgM into the efferent lymphatics so that it washes out of the lymph node and into the body and through the tissues. The IgG producing antibody cells, so the ones that have actually been through class switch recombination, often move to the bone marrow. So these are some of your longest lived antibody protection cells, and they're going to stay resident in your bone marrow for months to decades after your encounter with antigen, whether that's from a vaccine or whether that's from an actual infection. If the cells class switch to IgA because of conditions that are in the tissues, that tends to happen the most sort of at mucosal associated lymphoid tissues, and that's where these cells home to. So if they do get triggered in the lymph nodes to become IgA producers for whatever reason, those cells will then exit the lymph nodes and migrate to those mucosal associated lymphoid tissues. So like the gut associated lymphoid tissue or the bronchial associated lymphoid tissues in the lungs. The effector cells for T cells, the CD8 positive T cells in particular, travel to the sites of infection following chemokine cues. And these are going to basically also set up a kind of like a gradient that these cells are going to follow through the body. So they'll be able to sense and respond to increasing concentrations of these chemokines in the bloodstream. And then as they get closer to the site of infection and finally reach the site of infection, this is where those endothelial cells in the blood vessels actually express some different cell surface receptors, like the P-selectins that we saw in the first video when they were talking about leukocyte extravasation. So the cells will actually go through the rolling and adhesion and diapedesis and then migrate out into the tissues where they will perform their effector functions. Again, that's going to be perforin granzyme or fast ligand. The CD4 positive T cells, it depends on what kind of memory cell they become. So some of them are going to stay in the lymph nodes. Some of those T cells are going to go out and become, and those are the central memory T cells. And some of them are going to migrate back out into the tissues and to the site of infection where they become resident memory T cells as well. So this is kind of where we're at with our understanding. We've got kind of the two types of adaptive immune responses that go on during an infection, and that's the T cell response and the B cell response. And if you sort of separate these, the first initial thing that occurs in the early hours of a T cell response is antigen-presenting cell activation. That's step one, is going to be to make sure that your antigen-presenting cells actually acquire antigen from the environment, process that antigen, present it with MHC1 or MHC2, depending on the type of T cell that it is. Then those antigen-presenting cells, while they're processing the antigen and doing that, they're going to migrate to the nearest lymph node, where they will present that antigen to naive T cells and get that antigen into the lymph node. That part takes minutes to 24 hours or so, up to about 24 hours for antigen to be found in the lymph node, depending on the type of antigen and its size and its complexity. Over that first day of encounter between the antigen-presenting cell and the T cells, you start to get early activation events. So you get the formation of those immunological synapses as those two types of cells communicate with each other, the cytokine secretion that supports the maturation and then the proliferation of those cells. And the proliferation occurs usually between days about 1 and day 4 after initial antigen encounter. So this is an early kind of response, but it still takes 4 to 5 days. So we're still talking to up to 9 days after antigen encounter before these cells are really ready to migrate out into the peripheral tissues and attack the actual pathogen. That's a long lag period. Like, that seems to me like a long time to be unprotected. And this is why we have an innate immune response, is to protect us during this window where we're waiting for our T cell response to really gear up. And it's not that the B cell response is necessarily any faster, but that's kind of what's going on over on this other panel of this particular figure. So within the first few minutes, for soluble antigens, the small stuff, the lightweight stuff, that can be transported directly through the bloodstream and gets to the site of the lymph nodes or the spleen very, very quickly, so usually within minutes of entering the body. So the B cells pick up on those antigens within the first few minutes to the first 6 hours or so after antigen encounter, or during antigen encounter, I guess would be the way of kind of putting that. So around the 6-hour mark, you start to see B cells migrating to those T cell zones, and that's where we picked up today, was kind of where the B cells and the T cells migrated to those that central region between the follicles and the T cell paracortex of the lymph nodes, or the periarterial or lymphoid sheath of the spleen. At days 1 to 2 post-antigen encounter, what you see is B cell proliferation and migration still to that follicular zone. So this goes on from about 6 hours after antigen encounter for up to 2 days or so after antigen encounter. And by 3 or 4 days after antigen encounter, you still have cell proliferation, affinity selection, class switch recombination, all of those processes forming the germinal center. The shuttling between the light zone and the dark zone is still going on in there. And it takes a little while before you have a fully activated clone of really high affinity class switched antibody producing plasma cells come out of that. In the meantime, within 3 to 4 days, you start to see the development of the germinal center. So you've got plasma cells that are secreting IgM in the meantime that are lower affinity for that particular pathogen. And then starting in as little as 3 to 4 days after antigen encounter, you can start to see some egress or exiting of those effector cells from the lymph nodes into the tissues or into the bone marrow, or wherever those cells are actually supposed to kind of traffic to. So when and where do the memory cells come from, especially for the T cells, but also for the B cells? There are a lot of questions about this. Do these memory and effector fates occur early or late during the response? Is this like in the first division or after many divisions of these cells? Where exactly do they start to branch off? Do they get instructed to become memory cells by extrinsic signals? Like, is there some sort of cytokine or chemokine that tells certain cells that they should become memory cells? Or is this just sort of a random process? And these are questions that we don't have good answers to yet, unfortunately. So we don't honestly know. We do know that it takes about 7 days to start seeing effector and memory cells after a flu infection. So yay, that's normal influenza kind of kinetics there. That's a week after you've encountered the pathogen. Now keep in mind that during that week after you've encountered the pathogen, there's other things going on during that week on the pathogen side of this too. So it's infecting cells, going through replication, and your viral titers are still increasing during this first week of infection. You're going to have a few days where you don't even know that you've got the flu yet. So this is that asymptomatic period that precedes before you actually start to feel sick. Studies show that during this process, after about 9 or 10 divisions, a subpopulation of flu-specific CD8 positive T cells stops proliferating as quickly as the main population. So they slow down their rate of mitosis a little bit. And cells within this more slowly dividing population seem to be the ones that become central memory T cells on the CD4 positive T cell side. And they ultimately separate from the main pool of dividing cytotoxic T lymphocytes. So they seem to arise somewhere around those divisions 9 to 10 in that first week or so of proliferation. They sort of branch off from the rest of the T cells. And then the immune response starts to contract within 10 to 14 days. Like it barely seems to like get going, and then it starts to contract. Hopefully you've cleared the pathogen by this point in time. Otherwise, this contraction of the immune response can take a little bit longer. But this is due to two main things. The absence of proliferation cues. So as the infection winds down, you get less and less cytokine production, so there's less available to stimulate these T cells to divide, proliferate, and also to survive, really, in those peripheral tissues. And then you also get increased levels of like apoptosis-related factors that help to stimulate the onset of apoptosis in the immune cells themselves, not just in the tissues that were infected. So stimuli that contribute to the survival cues include the presence of antigen itself as a pro-survival cue for your leukocytes, in particular your lymphocytes, and cytokines like IL-2, which is necessary to drive a good, robust immune response, IL-7 and IL-15. After that antigen is removed, once you've cleared the infection, most lymphocytes are no longer required, and so they start to undergo apoptosis. And this is largely driven by T cells, targeting other immune cells for apoptosis. So the T cells express Fas, and activated T cells express Fas ligand as well. So you can get binding between Fas and Fas ligands, and T cells can kill other T cells. You also see a shift in cytokine production from these pro-inflammatory cytokines like IL-2, which is very pro-survival and stimulatory, towards CTLA-4 and PD-1, which we've seen before. These were major inhibitory cell surface receptors and markers that we used to determine whether cells were kind of pro-inflammatory or anti-inflammatory. In this case, we start to see an increase in the anti-inflammatory ones. Yeah. And it's basically about 95% of cells that actually will die off in response to a primary flu virus infection in the lung. So what we've kind of got are the kinetics, the timing and rate of events during this immune infection and when certain events are happening. And what you can see in this graph is that in the black line, you've got the virus titer and how much of that is present. And then you've got the CD4 positive T cell response, which you can see occurs earlier, a little bit earlier, and peaks a little bit earlier, not a lot earlier. But then you'll see a stronger CD8 positive T cell response. And this is in response to a viral infection. CD8 positive T cells are really, really necessary for clearing virus infections because they're the ones that actually have the ability to kill infected target cells based on MHC class 1 presentation of antigen. And then over those days post-infection, so starting around, let's see, the response peaks around day 10 or so, and then it gradually declines out to day 30 post-infection. So by that point, by about a month later, you should be well over this infection, and even your immune system should be basically pretty much back to normal. The exception here is going to be the memory cells. Some of these cells are still thought to survive and become ultimately some of your memory pool, some of those 5% that remain. So that's where we get to this section, which is kind of on the effector and the memory cell responses. Immune cells home to those sites of infection using chemokine cues and then addressins that are present in those endothelial cells of the blood vessels. So the addressins that we talked about earlier were specific for like the high endothelial venules and getting cells to migrate into the lymph nodes in the first place, getting the naive cells into the lymph nodes. These addressins are going to be slightly different. They're going to have different addresses. They're addressins. really not intended to be that bad and not intended to be like a pun or anything. It's just, I don't have a better word off the top of my head. It's kind of like having just a different spelling for a particular location that says, this is where you should exit. So what we've got going on in this diagram is a high endothelial venule and cells, naive T cells, so the T sub N's in the bloodstream, they're going to migrate into the lymph nodes, and then if they don't encounter their antigen, they'll upregulate the S1P receptor, which we previously learned about. They'll leave through the efferent lymphatics and circulate back into the bloodstream and repeat this process until you actually get an active productive infection. So that's what we've got kind of going on here, is these naive T cells migrate into the lymph nodes in response to these addressins at the high endothelial venules and in response to CCR7 and detection of CCL19 and CCL21. And then migration back into the blood vessel if they don't encounter their antigen. If they do encounter their antigen, they stop. So they don't continue to migrate. So these T-naive cells have entered, and then they go through an expansion. So you can see this nice cluster of T cells that have formed, and these differentiate into effector T sub E cells and memory T sub M cells. So you've got this cluster of cells that starts to form in the lymph nodes, and this is what causes your lymph nodes to swell up and become tender in response to an infection. It's not always something that's particularly notable because these cells don't actually stay in the lymph nodes. It's not like the lymph nodes become super enlarged most of the time, and they don't hang on to those cells. Those cells, their job is to then leave the lymph nodes and migrate to the site of infection in response to CD8 positive T cells in particular. But they have delayed exit and then they'll circulate back into the bloodstream, both the memory and effector cells. And some of those memory cells will re-enter back into the lymph nodes. If it's a central memory cell, it should stay pretty much in the lymph node. And others of those cells will migrate to the site of inflammation. Principally the T effector cells, but also your resident memory T cells will home to the sites of infection as well, and they'll pretty much stay in that tissue. So what they show in this particular diagram is this patch of the blood vessel over here next to all of these TE cells in the blood vessel, that stack of 3 TE cells. You'll notice that the wall of this blood vessel is a slightly different color. This is actually to represent that there's a different addressin here that is for CXCR3 and P-selectin, and E-selectin ligands. So this is a different addressin that is going to enable these cells to migrate out of the bloodstream and into this infected tissue, so that they can exert their effector functions. Okay, where do the cells go? Activated lymphocytes exit that lymph node and recirculate through various tissues. And depending, again, on the type of cell, it's going to end up in different types of tissues. So the 3 types of memory T cells, for example, the central memory T cells should stay in the secondary lymphoid tissues, the TCMs. The effector memory T cells should circulate among peripheral tissues and patrol and look for infected or reinfected tissues. And then the tissue resident memory T cells, the TRMs, are long-term residents of particularly in barrier tissues like the gut and the skin. And they help to compose some of those, what did IEL stand for? Isolated lymphoid, like the isolated lymphoid follicles of the gut, the isolated little patches of cells and that form these little clusters of lymphoid-like tissues that are present throughout the guts and the skin and also in your respiratory tract. So basically any of those types of tissues. The plasma cells, your effector B cells, home to peripheral tissues, including lymph nodes and bone marrow. So depending on the type of B cell, it's either pretty much just going to stay in the lymph node. For those plasma cells, especially the ones that don't really switch to IgG, and then the ones that do switch to IgG, those seem to be the ones that home principally to the bone marrow. IgA producers return to the barrier tissues, like at the gut and the lung-associated lymphoid tissues. For the CD8 positive T cells, those pretty much all migrate to the original site of infection where they kill cells that are infected that express MHC1 and peptide complexes. And that's pretty much where the different types of cells end up. So this is just kind of the graphical view of where these types of, where you find resident memory T cells throughout the body and where you find other types of memory cells throughout the body. So this includes, starting from the top and working our way down, you've got resident memory cells that are localized in the brain. You've got some that actually travel back to the thymus, in particular the medulla of the thymus, where they presumably participate in some of the training of T cells to make sure that they're not autoreactive against our own tissues. A bunch of them migrate to places like the lungs. A few go to the liver. Some go to the reproductive tract where they take up residence because, again, this connects usually to the urogenital tract. So having some protection and memory cells present in these tissues can be important. It should be very uncommon to get an infection that actually travels as far into the body as actually getting into the uterus itself or into the testes themselves. Usually we go to the doctor long before the infection can reach that far into the body. It's usually painful with very annoying symptoms that we would like to have gone as quickly as possible. So usually we seek treatment for that before there's a high risk of having those migrate to the deeper reproductive tissues. The lymph nodes, of course, are home to a number of memory cell populations in addition to the naive B and T cell populations, which primarily are what the lymph nodes are composed of. The intestines we've already talked about in the last chapter. There were a whole bunch of cells and cells involved, including memory cells that are going to be found scattered throughout that lamina propria. And in the skin, in the dermis, especially around the base of hair follicles. What's also interesting about the skin and the base of hair follicles is you also tend to find stem cells at the base of hair follicles. And it's one of the pools where we can actually isolate adult skin stem cells that can turn into any kind of skin cell is actually from the base of your hair follicles. You look like that's gross. That's gross? There's usually not like a huge pool of them, and it's not like every hair follicle on your body, like the ones on your arm. It's not like all of them have stem cells at the base, but a good number of them actually do. So it's really about the chemokine receptors and integrins that regulate homing of these effector cells to the peripheral tissues, especially like the CD8 positive T cells, because those are the ones that mostly migrate into the infected tissues in response to an active infection. Most of your plasma cells either stay in the lymph nodes or migrate to the bone marrow and continue to pump out antibodies that get entered into the bloodstream. But these effector cells actually downregulate L-selectin, and one of the things that does is it prevents them from re-entering the lymph nodes through the high endothelial venules. That keeps them from recirculating and homing back to the lymph nodes. So instead, they express adhesion molecules and chemokine receptors to home to these different sites of infection, and that's part of what's being depicted in these two panels of this figure. Is you've got one cell that's a mucosal homing effector T cell and one that's a skin homing effector T cell. This is how the cells actually find different tissues and migrate into those different tissues. So for the mucosal tissues, you've got some L-selectin and some MADCAM that's necessary for migration into the mucosa. ICAM1 is also important for migration into the mucosa, and LFA1, which is present for both skin homing and mucosal homing cells. It's just kind of a generic intracellular adhesion molecule receptor, so it's not particularly specific for ICAM1. Well, it may be specific for ICAM1. It's shown in both of these panels. But in the skin homing cell, you've got a different receptor binding to a different antigen on these skin dermal cells. So this is basically what we mean when we're talking about the addressins. The T cells moving to the intestinal mucosa express things like LPAM1, LFA1, and CCR9, whereas the ones going to the skin express a different subset of molecules, which are CLA, LFA1, CCL17, CCL27, and CCL1, and those are mostly expressed by keratinocytes in the skin. So your skin cells are active participants based on the chemokines that they make, which are going to be in response to the pattern recognition receptors being triggered and activated. They'll start making these different cytokines and chemokines that basically enable the effector T cells to home into these particular tissues. Okay, and in the last section of this particular chapter, some of the case studies and some of the interesting specific examples of immune responses to particular pathogens. The first one is near and dear to my heart because it involves cats, and yes, I am a cat mom. I'm definitely a crazy cat lady. I like dogs too, but I have cats, so I don't have actually any dogs at the moment. But this one is Toxoplasmosis gondii, is the pathogen. And this one is actually transmitted to cats and humans, from cats to humans specifically, and other animals such as mice via the feces, which is why they do not recommend that pregnant women scoop litter boxes. There's actually a specific prohibition against pregnant women scooping cat litter boxes. And this is because this particular pathogen doesn't really cause any symptoms or problems in adults, but it can in neonate and developing embryos. So it actually specifically will travel to the brain. So it forms what you get from the litter box basically are oocysts, which get into the body and can form tachyzoites. These tachyzoites are rapidly dividing little cells that can actually enter into the brain, where they can set up cysts and just kind of stay resident in the brain. This actually changes the behavior of the host animal. So in particular, in response to toxoplasma, the behavior includes reduced fear of cats. I probably have this parasite. Actually, estimates are that something between 50 and 90% of all humans have this parasite. So you probably have this parasite too. Congratulations, that's so gross, but it's reality. But what's interesting is that in response to this particular parasite, mice lose their fear response to cats. So mice don't run away from cats anymore if they've actually been infected with Toxoplasma gondii. Mind control pathogen there. And they can also show behavioral changes in humans, such as increased feelings of affection towards cats. They think is one of the things that underlie crazy cat lady syndrome. Guilty as charged. So, let's see, I've already kind of covered some of that. The CD8 positive T cells in the lymph nodes respond within 36 hours of infection and form stable contacts with dendritic cells that last for up to 8 hours and peak at 48 hours. So the first 2 days after infection, you start seeing activation of the CD8 positive T cells. The responding CD8 positive T cells and dendritic cells collect in the subcapsular sinus of the lymph nodes, which is what we've all been talking about so far. The subcapsular sinus is going to be important because this is where you're going to have a lot of those immune cells. And one of the cell types that actually gets infected by toxoplasma are macrophages. So when macrophages attempt to phagocytose these particular pathogens, they actually become infected themselves. And then they can spread this infection throughout the rest of the body through the circulation, and by passing those infected macrophage bits can actually spread the infection to T cells themselves and produce infected T cells. So this is why clearing a toxoplasma infection is really, really difficult, because the pathogen itself can actually hijack the immune system and use it as a method to travel throughout the body. Lymphocytes entered the brain starting on day 3 post-infection, and again, this can be carried in, like our own immune system can actually work against us in this particular case. So these lymphocytes entering the brain by day 3, some of those may actually be infected and be carrying the infection into the brain, and it peaks on about day 22, which coincides with the proliferation of the CD8 positive T cells in the lymph nodes. So not all responses wind down after 10 to 14 days. Some of them go on considerably longer, such as the one to Toxoplasma gondii, and around day 40 we start to see a problem, and that's a loss of function associated with the upregulation of PD-1 for negative co-stimulatory molecules. That starts to occur, and this decreases the ability of T cells to respond to the infection. So around day 40, it's not really the contraction phase of the immune response, it's actually more like a fatigue of the immune response. It just can't respond as strongly or robustly anymore. Those T cells are expressing some inhibitory molecules, and this exhausts the lymphocytes and reduces our ability to productively or to actually clear the infection. Another really good one is the herpes simplex virus, HSV. So herpes simplex virus is part of the herpes family. We all probably have at least one strain of the herpes virus family infected in our bodies in some way, shape, or form. Herpes viruses, I think, are like the pinnacle of evolution because they don't really make you sick. They give you like a cold sore or a fever blister, but that's not enough to keep people home from work. So you go out into the general population where you can spread your infection to other people, where it will similarly not make them super sick. They're really highly evolved little pathogens that do a good job of spreading throughout populations without causing a lot of illness or particular damage. So they're not super virulent. They rarely cause major health problems. Again, it's usually in neonates, so babies right around before and at the time of birth that are the most susceptible to herpes simplex type complications, especially from something like LCMV, lymphocytic choriomeningitis virus. But in this particular case, HSV infections are sometimes cleared within hours. So usually the innate immune system catches these and clears these infections. But for those that are not, infections can become latent. And what that means is that in a latent infection, the virus and the viral genome gets incorporated into the host cell's DNA. That means it becomes part of you. It is now yours. It cannot be separated back out. So the only option is going to be to kill that particular infected host cell altogether. There's no way to trap and eradicate that virus from that cell. You just have to kill the whole cell. And that can persist for the lifetime of the host. Herpes viruses are not the only types of viruses that do that. Vesicular stomatitis virus does not, but the one that causes chickenpox, varicella virus, does. So it can remain resident in some of your nerves in particular and reactivate later in life, causing shingles, which is incredibly painful. That's why there's a shingles vaccine, and they will recommend that you get that once you're like 65 and older. That's to boost your immunity and help to make sure that those viruses do not reactivate. But these are particularly difficult for the immune system to deal with because the virus can effectively hide for long periods of time. And when I say long periods of time, I mean years to decades. So people who get shingles primarily had chickenpox when they were kids. And most of you are young enough that you got vaccinated against chickenpox. You never even had chickenpox. There's a couple of you in here that might have actually had chickenpox. I had chickenpox. It sucked. I was like 4 years old. I don't really particularly remember it, but mom said I had a really bad case of it and I was super itchy and it was very distressing to her to have a baby that was that miserable for that period of time, because this lasts for a couple weeks. You get these pustules and these sores that really are super, super intensely itchy, and scratching them can cause scarring. So you want to keep your kid, your 4-year-old child, from scratching an itchy thing. Good luck with that. One way to do that is by putting socks over the arms, like pinning them to the t-shirt sleeves. That works well, but it's not a fun particular infection to have. It's also not particularly lethal. Kids do actually die from chickenpox, but it's rare. So herpes simplex, most people do not die from herpes simplex viruses. They just cause resurgent occasional symptoms to flare up, which are usually uncomfortable, and that's usually, and maybe like ugly, and that's about it. So what we're looking at in this particular case is the fact that about 2 weeks after infection, about 5% of T cells are actually positive and specific for HSV, herpes simplex virus. And these T cells actually also show a different morphology than we expected. So if you look at them and you look at the pictures real quick, you'll notice that they are not round little lymphocytes like they show in the textbook illustrations. These are highly branched cells that look more dendritic cell shaped in morphology. So this seems to be kind of a consistent theme for the immune system. In terms of infection and reactivation of existing infections, the resident memory T cells are the first responders to herpes simplex virus reactivations and reinfections. So you're relying on those memory cell responses to really kind of help bring those reinfections or reactivations of existing infections back under control. And they release inflammatory cytokines, including interferon gamma, on antigen engagement. So that helps to drive something that looks like a normal secondary immune response. And these cells can respond within hours. These cells are dendritic-like in shape, but they're also continuously mobile. So they'll actually like migrate through tissues and look for antigen. And then once they encounter antigen, they'll rapidly start to have a memory cell response. They'll start to proliferate and secrete cytokines, and that will help to generate some effector cells that can help control that infection. So in this particular video, one of the things that you're going to see are some of those HSV skin resident T cells, and what you're looking at are the resident memory T cell pool in the skin. The cells are green fluorescent protein positive, so that's why they're glowy. And in this particular case, notice how these resident memory T cells slow down and then round up. So they're actually going to stop being quite so dendritic-like in morphology and turn into these more round, classic, like what we think of as a lymphocyte-looking cells. So the cells moving around in the skin, this is vaguely creepy. Like, I don't really like to think about like a whole lot of movement going on in my body under my skin. That's disturbing, but these cells are constantly kind of crawling around and patrolling in your skin. And then once they encounter antigen, the cells actually stop and get more round in terms of their shape. But as far as I can tell, there's not antigen reintroduction in this particular video. It's just looking at the basic movements of these resident memory T cells. And you wouldn't think that in a particular patch of skin there would be that many of them. So this must be a particular area of skin where there had previously been something like a cold sore or a fever blister. So there would be a higher population of these resident memory cells in that particular tissue and in that particular location in specifically. Okay. So grafted tissues are another interesting immunological case. This has to do with tissue transplant in particular for humans. But we do this mostly in studies with mice because mice are easy to manipulate in the lab, and you can do this with something as simple as an ear punch. So the role of host versus donor antigen presenting cells is not well understood. So the dendritic cells that we talk about in terms of being the drivers of our immune responses, the first question you kind of have to ask in a transplant situation is, which ones? The ones from the host or the ones from the incoming transplanted tissue? Because when you transplant a tissue, you're also getting some small number of antigen presenting cells that are resident in that tissue, and also some other small resident number of immune cells that are probably going to be resident in that particular tissue, even if you're talking about something that's kidney instead of skin patches. So what we do know is that what we've got in this particular case, the rejection of grafts that are MHC mismatched, so allografts, requires activity from both CD4 and CD8 positive T cells. You've got to have both types of cells. And again, this goes back to getting optimal activation of those CD8 positive T cells, probably with those trios. Both types of the T cells have to interact with the antigen presenting cells that are going to express the graft's antigens, the foreign antigens. So in this study, a mouse whose dendritic cells are yellow, that's what we've got in the particular panel of this particular image, was used to track the motions of antigen-presenting cells. And these fluorescent yellow cells exited the graft and were gone within 6 days. They show up then in the draining lymph nodes of the tissue. And this starts within about 3 days after engraftment. So the first 3 days, basically, the resident dendritic cells migrate out of the tissue and toward the nearest draining lymph node. And that's kind of an interesting observation. But what's also really interesting about this is that those dendritic cells didn't seem to do anything once they got to the lymph node. They actually, basically kind of rounded up and died off. They didn't seem to persist. They didn't seem to particularly do well. They didn't remain healthy. So they just become kind of orphan cells that can't really or don't seem to do much. Could they be triggering an immune response in those lymph nodes? Yes, they could, but we're not sure if they are or not, and that's one of the things that we're still trying to tease apart. They also tracked some myeloid cells from the mouse that were GFP positive, and noticed that those infiltrated into the graft also within 3 days. This was at first dominated by neutrophils. Neutrophils are always the first responders to your infection, but they're also your first responders after surgery. So to any sort of trauma or inflammatory response, it's usually neutrophils first. And these come into the MHC matched transplanted tissues as well. So it doesn't have to be a foreign tissue. Even if you took a patch of tissue from one area of skin and transplanted it to another area of skin, you would see neutrophil infiltration into that moved patch of skin. So, and this is probably just due to inflammation and inflammatory reactions from the surgery itself and the damage that's associated. So that goes back to the damage-associated molecular patterns that we talked about way back at the beginning of the semester, the DAMPs. We do know that cells from the host, especially antigen-presenting cells from the host, will migrate into the grafted tissues as well, and they actually will stay in those tissues and appear to do well, and we actually see infiltration of lymphocytes, CD8 positive T cells. Usually by day 10, they're found throughout the graft in all 3 tissue layers. So that suggests that the host is definitely, that's a tissue that's going through active rejection at that point in time. That would be the green cells in the second panel, the CD8 positive T cells that are actually kind of present in that tissue. So you've got a few little scattered CD8 positive T cells here. One of our newer faculty members, Dr. Rishi Drolia, who just came to us and started basically this semester here at ODU, he does his research on Listeria monocytogenes, which is a really interesting pathogen. It is actually really good at evading the immune system because it forms intracellular infections. So it actually it's a bacterial infection, but it gets inside the host cells. And it actually forms these really interesting little comet-like tails that are due to reorganization of the actin cytoskeleton. So what you see is the actin tails produced by Listeria in green, and the infected cells, the red cells, are actually the Listeria bacterium themselves. And then I've got a video that's in the bottom panel that I'll play in just a minute. But this is a pathogen that is found principally in soil and food. So you've got the tails and then the bacteria, the little head pieces. And it can cause serious illness. And it can actually be fatal. I don't think it's commonly fatal in this country, especially because we have good treatments for that. But these bacteria can actually escape into the immune system through the dendritic cells, where they can use those cells as vectors to get through to the rest of the body. So within minutes, the listeria get phagocytosed by dendritic cells in the red pulp sinuses of the spleen, and that infects the dendritic cells. And now you've got infected immune system cells again. And so the immune system then has this difficult task, killing itself for the sake of the host, which it is not designed to do. This is why it's so hard for us to eradicate HIV. HIV was first detected back in the early 1980s. It's something that was scary as shit when I was a kid. Like, you wouldn't believe the things that we were actually taught, but we were taught not to even shake hands with other kids because they might be HIV positive. We were that afraid of it, and we didn't understand well how it was transmitted at that point in time. So we didn't realize that it was transmitted primarily through blood and blood products, and not even really through saliva. Like, the risk from saliva is extremely low. But this idea that the immune system then has to kill off some of its own types of cells is completely alien to the way the immune system works. It does not usually target itself as a way to eradicate infections. So the other thing that's interesting, and this video is the one that I'll play now, actually kind of takes us through what happens with these bacteria as they infect a cell and how the infection spreads from cell to cell. So just imagine this is a cell infecting a dendritic cell.
This mammalian cell has been infected with pathogenic Listeria monocytogenes.
So you can see those
These
actin
bacteria
tails.
move throughout the cytosol by recruiting host cell actin, which polymerizes and pushes them forward, producing a comet's tail in their wake. These bacteria can also form actin comet tails and move in cell extracts. Here, the bacteria are expressing the green fluorescent protein, and actin is labeled red with a fluorescent dye. Whenever a bacterium is pushed into the plasma membrane, it creates a temporary protrusion and is then bounced back to continue its random path. Occasionally, instead of being bounced back, the protruding cell membrane and the listeria within it are phagocytosed by another cell. When this occurs, the Listeria bacterium is able to break through both sets of cell membranes and enter the cytosol of its new host cell. Once in the cytosol, the Listeria is able to replicate rapidly. In this way, the bacterium can spread from cell to cell without ever entering the extracellular fluids, effectively hiding from the antibody response.
So that becomes a problem because the CD8 positive T cells themselves are the prime targets for these Listeria bacteria. They'll infect dendritic cells, but they use those dendritic cells as a stepping stone to get to the CD8 positive T cells. In much the same way, HIV actually infects CD4 positive T cells and hides from the immune system that way. And that is apparently where I get to stop for today because we are out of time.
This mammalian
And this last couple slides actually was looking at the T cell response to tumors as an example. And one of the things that we know and that we understand is that the immune system should be scanning for antigens that don't look right. And whether that's a foreign antigen or a messed up native antigen, one that's actually, endogenous to the body, shouldn't make a huge difference. T cells should be able to recognize and clear those tumor cells. And we know that T cells can do this based on in vitro studies. So what you're looking at in this image is actually a tumor on the kind of upper and right side of this white squiggly line, and the surrounding tissue with the yellow cells on the bottom and left side of the image. And you can see that there's more T cells in the surrounding tissue, but a few of those T cells actually do penetrate into the tumor. So we know that T cells actually get into tumors and cancers and attack those tumor cells and can actually kill those tumor cells and induce apoptosis. However, in this particular case, as in many cases, the CD8 positive T cells alone were not sufficient to clear the tumor. So the cancer did not get cured or completely removed by those T cells, but it didn't really even regress. However, this idea is central to CAR T therapy for cancers. This is where a patient's T cells are harvested from a blood sample grown up in a lab and treated to ensure that they recognize a particular antigen of a specific kind of cancer or a specific kind of tumor, and then re-injected and reinfused back into the patient. So you literally get your own T cells, just a big bunch of them that have been specially grown in the lab and trained to recognize the particular cancer that you have. This treatment mostly appears to be used for blood cancers, so like leukemias and lymphomas. T cells have a hard time penetrating into the middle of an established large, especially solid tumor. So that's something that is an active area of investigation, is how can we make it easier for T cells to actually get into the tumors and make it more likely that they survive in those tumor microenvironments so that they can actually do their function and produce apoptosis in the tumor cells and hopefully kill off the cancer. This would be very helpful to our ability to treat and cure cancers. But so far, we have not made a lot of inroads on that. And last but not least, the regulatory T cells can actually inhibit the immune response in multiple ways. So in a model of type 1 diabetes, there's a role for regulatory T cells. They can inhibit effector T cell function by quelling activation and the potential of resident antigen presenting cells. So they help to tamp down on the autoimmune cause of type 1 diabetes. And they can form stable connections with effector CD8 positive T cells, inhibit their killing capacities by secreting one of the two major anti-inflammatory cytokines, TGF-beta. And these findings are still consistent with the modes of action of regulatory T cells. We know from our studies in the gut mucosa and from elsewhere in the body that these are generally inhibitory cells. They often secrete and respond strongly to IL-10 and TGF-beta, so this is not really surprising. But it's interesting that they actually found these active in a model of type 1 diabetes. And so that's literally the end of chapter 14. We have now finished that up. Cool.